Welcome to Clinical Histology. I am Dr. Xie from SUMC. We will go over smooth muscle in this session. By the end of this session, you are able to define a smooth muscle, describe a dense body and a dense band, and explain how smooth muscles work within internal organs and passageways throughout the body. Smooth muscle is characterized by its slow, steady contraction, regulated by autonomic nerves and by various hormones or microenvironmental factors. Hence, smooth muscle is an involuntary muscle. Take the intestinal smooth muscles as an example. People do not need to think about their small intestine for its peristaltic activities. The spontaneous movement of the intestinal smooth muscles, called peristalsis, are controlled by neural impulses and hormonal factors. Smooth muscle is found throughout the body, such as the walls of hollow visceral, blood vessels, and respiratory tracts. Without normal activities of smooth muscles, the organs and systems in our body would not be able to maintain even their most basic functions. Now, let's take a look at these two H&E sections. On the left is a longitudinal section of smooth muscle. On the right is the cross section. In both sections, a single round nucleus is centrally placed in each cell, which is similar to cardiac muscle, but different from skeletal muscle. Smooth muscle fibers are spindle-like with no striations. Upon contraction, smooth muscle shortens and acquires a characteristic corkscrew shape. Individual smooth muscle cell is surrounded by endomycin as well, which along with other connective tissues hold the cells together and combine the contractile force produced by many smooth muscle cells to generate a coordinated action, such as intestinal peristalsis. Smooth muscle cells in specific organ are often organized into layers with various thickness. Neighboring smooth muscle layers are usually oriented at right angles to each other. This arrangement of smooth muscle layers is another structural basis for coordinated actions at organismal level. Now, let's move on to fine structures of smooth muscle cells. On the sarcolemma of smooth muscle cells, we can see lots of small invaginations that make the membrane extremely wavy, as indicated by red arrows in the micrograph. These invaginations are known as caveoli. Smooth muscle cells do not have T-tubules. Instead, they have T-tubule analogs, the caveoli, which play the roles of T-tubules in skeletal and cardiac muscles. In this electron micrograph, we can easily identify the nucleus on the upper left. Just beside the nucleus, you can see the mitochondria, polyribosomes, RER, and vesicles of a Golgi apparatus. The most obvious structure in a cytoplasm is the dense body. They are electron-dense thick lines in the cytoplasm, as indicated by the red arrow. You can also find similar electron-dense lines just beneath the sarcolemma, indicated by the green arrow. They are so-called dense bands. Dense bodies and dense bands function in the same way, like the Z-discs in skeletal and cardiac muscles. They provide anchoring spots for thin filaments and other cytoskeleton components, such as intermediate filaments. They transmit the contractile force not only within, but also between cells. In a cytoplasm, there are arrays of filaments, including the thin and thick filaments. they behave similarly to that of skeletal and cardiac muscles, and set the foundation for contraction in smooth muscles. However, they are not organized into sarcomeres. Thus, there are no striations and the muscles appear smooth. That's why they are known as smooth muscles. The termini of thin and intermediate filaments are either anchored to the dense bodies in cytoplasm or to the dense bands on plasma membrane, as shown on the left. They form a network in which the dense bodies and the dense bands serve as the nodes. As mentioned above, at the basic level, smooth muscle contraction also rely on the sliding of thin and thick filaments, just as in the skeletal and cardiac muscles. As shown on the right, the force generated by the sliding of thin and thick filaments drags the dense bodies or dense bands closer to each other. The myofibrils in smooth muscles are arranged into networks, rather than uniformly aligned like those in the skeletal or cardiac muscles. Therefore, when a smooth muscle contracts, the forces squeeze the cell from many directions, and as a result, the cell becomes corkscrew-shaped upon contraction. There are two types of smooth muscle. One is single-unit smooth muscle, and the other is multi-unit smooth muscle. Cells in multi-unit smooth muscle contract independently to each other because each cell receives its own neuron input, and there is almost no gap junctions between them. This type of smooth muscles is found in the walls of large blood vessels, respiratory passages, and in eyes. In contrast, the single-unit smooth muscles are connected by gap junctions, as depicted in the lower picture. Thus, upon stimulation, the cells in single-unit smooth muscle respond simultaneously and contract synchronously or in a coordinated manner. This type of smooth muscles is found in the walls of visceral organs except the heart and are commonly called visceral muscles. Asthma is a chronic inflammatory airway disease. Patients who die of an acute severe asthma attack have 2 to 3-fold increase in amount of bronchial smooth muscles. The chronic inflammation will also result in thickening of the smooth muscles, as indicated by the yellow arrows on the right. Smooth muscle contraction leads to a decrease in the diameter, that is, constriction of the bronchioles. In patients with asthma, the smooth muscles that encircle the airway undergo prolonged constriction, limiting the flow of air to and from the lungs. This, therefore, makes the patient feel breathless and tight in their chest. Drugs such as beta agonists will relax the smooth muscles in the airways and relieve asthmatic symptoms. For this session, we have talked about the structural features of smooth muscle. Smooth muscle cells are small spindle-shaped cells. They have no sarcomere, thus no striations. Instead, they have functional analogs of Z-discs, the dense bodies and dense bands. They have no transverse tubule system, but they have caveoli, which act similarly to the T-tubules. Although thick and thin filaments also exist in smooth muscles, they are not arranged in myofibrils. The key to understand smooth muscle contraction is the network mesh of filaments, connected by dense bodies or dense bands as the nodes. The sliding of thin and thick filaments in various directions is the main cause for the corkscrew shape of the smooth muscle upon contraction. That's all for today. Thank you for joining us. See you next time.